Good. Okay. So, hi. It's a pleasure to talk to you today. So, what I'm going to do is give you sort of a brief overview of angiogenesis, invasion, and metastasis. And the reason why I say I'm going to give you a brief overview is because these are massive subjects. I would normally, in the module I convene, spend 3 hours on them minimum. So condensing this down to 1 hour, hopefully I can give you a flavor for the importance of both angiogenesis and invasion and metastasis in tumor progression towards malignancy. So, slides aren't progressing for some reason. Okay, here we go. So, I'm sure you've all been introduced to the hallmarks of cancer, the seminal review by Hanahan and Weinberg. Initially, there were 6 hallmarks, and further have been added in the intervening years. But inducing angiogenesis is one of those original 6 hallmarks. And these hallmark characteristics are characteristics that a tumor has to acquire in order to progress to become a malignant cancer. So, inducing angiogenesis. So, what is angiogenesis? It is the production of new vessels, either blood vessels or lymphatic vessels, from the existing vasculature. And tumors require the induction of angiogenesis to be able to grow to a large size. So, just like a normal tissue, a tumor requires access to the vasculature to give it access to oxygen, nutrients, to be able to shed metabolic waste products. And without this access to the vasculature, they will suffer from hypoxia and die. But it's important to stress that angiogenesis is not required for tumors to form, and it's not required for tumors to grow. So a tumor can form and it can grow to a certain size without the need to induce angiogenesis. And that's because at a small size, a tumor can make use of the existing vasculature that's already within the tissue. But once the tumor grows beyond a certain size, the cells in the center of the tumor start to get further and further away from the existing vasculature, and that's when they start to suffer from hypoxia. So you might have a very highly proliferative tumor, but if it hasn't triggered angiogenesis, there'll be a lot of cell death there as well, and so the tumor mass won't increase dramatically. It's only with the induction of angiogenesis that a tumor can grow to a large size. So here I've got an image of a human ductal in situ breast carcinoma. The tumor is labeled in blue, and in brown you can see a blood capillary, and you can see a branch coming off that blood capillary and surrounding the developing tumor, supplying everything that tumor needs to grow to a large size. So how do tumors induce angiogenesis? Well, they can secrete pro-angiogenic factors. And this table shows the wide variety of pro-angiogenic factors that can be secreted, but by far the most common is vascular endothelial growth factor, or VEGF. And you have different VEGFs, and they bind to different receptors, and they can induce both blood vessel angiogenesis and lymph angiogenesis. And so a tumor, depending on which VEGFs it's secreting, can promote either blood vessel angiogenesis or lymph angiogenesis, or both. So the tumor will secrete these pro-angiogenic factors, but in order to induce angiogenesis, those pro-angiogenic factors have to reach the existing vasculature to allow a new vessel to grow towards the tumor. Now, the problem the tumor has is that when it secretes these pro-angiogenic factors, it secretes them into this. So here you can see a scanning electron micrograph, and it shows epithelial cells, and beneath those epithelial cells is the basal lamina, and beneath that is the extracellular matrix. And you can see how the extracellular matrix is this tight mesh of long protein fibrils. And so what happens is that these pro-angiogenic factors can be secreted by the tumor, but very often they get stuck, they get sequestered within that extracellular matrix. And So what the tumor can do is not only secrete the pro-angiogenic factors, it can also secrete lytic enzymes called matrix metalloproteases that can break down that the protein fibrils and allow the pro-angiogenic factors to move further away from the tumor. So what this does, the degradation of the extracellular matrix, it has two major consequences. One major consequence is very relevant when it comes to invasion and migration, in that it leads to a breakdown of the extracellular matrix, a structural remodeling. And obviously, if you have cells that break off the primary tumor and invade and migrate away, they're going to find it much easier to migrate away when the extracellular matrix has been degraded rather than through this very, very tight mesh. The second consequence is more relevant to angiogenesis itself, and that it allows the release of a variety of different factors that might have become sequestered within the ECM, and that can include VEGF, but also things like growth factors and survival factors as well. So you've got the tumor secreting these pro-angiogenic factors and these matrix metalloproteases, but very often even that is not sufficient for the angiogenic factors to reach the existing vasculature. So what it also does is it recruits other cell types, such as cells of the immune system. It recruits them and also induces those cell types to also secrete both pro-angiogenic factors and lytic enzymes. And it's this combination of the tumor and the surrounding stroma secreting these factors that allow these angiogenic factors to reach the existing vasculature. Once those factors reach the existing vasculature, they will bind to the VEGF receptors on the surface of the blood vessel or the lymphatic vessel, and that will lead to a branching of that vessel and a growth of a new vessel towards the tumor. And it will grow directly towards the tumor because that vessel will grow up the concentration gradient of those VEGFs. And obviously VEGF is most concentrated in and around the developing tumor. Now I want to stress how this type of angiogenesis or lymphangiogenesis is very different to what would normally happen under physiological conditions in a normal tissue. So in a normal tissue, you've got this balance of pro-angiogenic and anti-angiogenic factors within the tissue. And if that tissue requires the formation of a new vessel, then what happens is that you slightly tip the balance towards the pro-angiogenic factors, and that will lead to the slow and deliberate formation of new vessels at the right time. However, this doesn't happen in a tumor environment. What happens in a tumor environment is you get this flood of pro-angiogenic factors, and that leads to very rapid formation of new vessels. And these rapidly formed vessels are very poorly formed, they're immature, and they're very, very leaky. So here you can see an example of the vessels in a normal tissue and also in a tumor environment. You can see this beautiful organization, beautiful hierarchy of blood vessels and blood capillaries in normal tissue. And you compare that to a tumor environment and you've lost that beautiful hierarchy, you've lost the organization. Instead, you have a chaotic organization, and these rapidly formed chaotic vessels are very poorly formed and very leaky as a consequence of it. And it's also known that high concentrations of VEGF can also induce further capillary permeability. And this is where angiogenesis is really linked to invasion and metastasis. So once a tumor forms and it becomes invasive, a cell will break off that primary tumor and it has to migrate away from that primary tumor. And in order to form a metastasis at a distant site in the body, it makes use of the vasculature. So it intravasates into a blood or lymphatic capillary, and then that capillary, the vascular system, sort of acts as a superhighway. It can transport these migrating cancer cells all over the body, and it can transport these cells to distant sites, and that facilitates the formation of multiple secondary tumors at multiple secondary sites in an individual. Now, angiogenesis promotes the formation of these metastases in a number of ways. So first of all, we spoke previously about how the matrix metalloproteases lead to the breakdown of the extracellular matrix. So a cell breaking off that primary tumor will find it much easier to migrate away when the extracellular matrix has been degraded. Secondly, if angiogenesis has been triggered, it means that a vessel has grown towards the tumor, which means that when a cell breaks off that primary tumor, it doesn't have to migrate very far before it encounters a blood vessel or a lymphatic vessel. Thirdly, these vessels are very poorly formed. They're very leaky, which means that it's relatively easy for these migrating cancer cells to actually infiltrate and enter a blood vessel or a lymphatic vessel. And fourthly, as I said previously, if you have high concentrations of VEGF, that increases the permeability of these vessels and makes it even easier for these vessels, for these migrating cancer cells to intravasate into these vessels. So that brings me nicely on to the second hallmark that I'm talking about today, which is activating invasion and metastasis. Now, this probably is the most important hallmark when it comes to cancer, because it's the hallmark that separates a benign tumor from a malignant cancer. Benign tumor is self-contained, a malignant tumor is invasive. And so this hallmark separates basically a true cancer from a benign tumor. So what this slide here is showing is the basically a schematic of tumor progression. So you start off with cellular transformation, you have a mutation in an oncogene or a tumor suppressor gene that leads to uncontrolled cell proliferation. And that leads to the formation of a benign tumor. Now, benign tumor is characterized by the fact that it's self-contained, it's not invasive, but also cells within the benign tumor more or less maintain their normal characteristics of the tissue of origin. So here we're talking about a columnar epithelium. So cells within this tumor will maintain their normal columnar cell shape. They'll maintain their junctions with their neighbors. They'll maintain normal contacts with their neighbors and with the underlying basement membrane. But as that tumor acquires more and more mutations, as it moves further and further down that road toward malignancy, that tumor eventually becomes invasive. And when it becomes invasive, it starts to lose these normal characteristics of their tissue of origin. So here the cells will lose their normal cell shape. They will break their junctions with their neighbors. They will break through the basal lamina. They will lose normal cell polarity, and I'll go on to talk about polarity in a moment. And that will promote invasion and eventually migration away from the primary tumor. And what I've just described to you is a process known as epithelial to mesenchymal transition, or EMT. So epithelial to mesenchymal transition is the loss of epithelial characteristics and the gaining of mesenchymal characteristics. And this is important because the vast majority of cancers are epithelial in origin. So this is relevant to the vast majority of cancers. So epithelia have a number of characteristics, and one of those are cell-cell junctions, and the most characteristic junction of an epithelial sheet is the adherens junction. And at the core of the adherens junction is a protein called E-cadherin. And E-cadherin forms very, very strong junctions between cells. So neighboring cells sort of stick together. It's the glue that maintains epithelial integrity. Another characteristic of epithelial cells is a type of cell polarity called apical basal polarity. So cell polarity gives a cell spatial information, and apical basal polarity tells a cell where its top and where its bottom is, where its apical side and where its basal side is. And this is important for a cell to correctly position subcellular structures, proteins, lipids, etc. Another characteristic of epithelia is a robust cell shape. So epithelia have characteristic robust cell shapes such as columnar or cuboidal or squamous. Now, when a cell undergoes EMT, it loses these epithelial characteristics and instead takes on mesenchymal characteristics. So instead of the cell expressing E-cadherin, forming these strong junctions between neighbors, instead it expresses a protein called N-cadherin. And N-cadherin doesn't form strong bonds between neighboring cells. Instead, it promotes transient weak interactions. Mesenchymal cells also have a different type of polarity called front-rear polarity. So here, instead of the cell knowing where its top and where its bottom is, it knows where its front and where its back is. And that's really important for promoting directed migration. Thirdly, it doesn't have a robust cell shape. Instead, it has sort of a spindly type of shape, a fibroblastic type shape. And all of these things promote cell motility. So the transient weak interactions instead of strong junctions, the front-rear polarity, the spindly type shape, all of these promote cell motility. And another thing that happens when a cell undergoes EMT is it starts to secrete matrix metalloproteases. So we spoke about those previously. And I'll show you this again. So again, this is showing the epithelial sheet. Beneath the epithelial sheet, you can see the basal lamina, this very thick, mat-like substance composed of laminin that the epithelium has secreted. And you can see how this very thick sheet underlying the epithelial sheet a cell wouldn't simply be able to push through this basal lamina. It has to, in order to invade, it has to actively degrade that basal lamina using those matrix metalloproteases. And once it does invade and break off that primary tumor, then it finds itself in the ECM. And as you can see, the ECM is composed of these very, very long protein fibrils that can be very tightly knitted together. So again, it has to continue to secrete these matrix metalloproteases to clear a path through which those migrating cancer cells can migrate away from that primary tumor. So in epithelial to mesenchymal transition, cells lose epithelial characteristics such as cell-cell junctions, such as normal robust cell shape, such as apicobasal polarity, and instead they take on a spindly type of cell shape. They take on a front-rear polarity, and they lose junctions. They break their junctions with their neighbors, and instead are only promoting transient interactions. Now, I just wanted to take a moment to highlight the importance of the adherens junction and how it is regulating, but also regulated by other processes in the cell. And so here you can see the adherens junction, and the core of the adherens junction is a complex known as the cadherin-catenin complex. So the extracellular domain of E-cadherin is the part that binds and sort of glues cells together within the epithelial sheet. But the intracellular side of the protein interacts with a number of different catenins, and that links the junction to both the actin and microtubule cytoskeleton. And these catenins can also interact with a number of actin regulators, so that the junction can regulate the actin cytoskeleton. Now, the junction is also important in establishing and maintaining apicobasal polarity, the thing I spoke about previously, that allows the cell to know where its apical and basal sites are. So what this means is that if you lose E-cadherin expression, for example, it's very common in a human cancer for E-cadherin expression to be lost. Not only do you disrupt the adherens junction and you break junctions between neighboring cells, you also disrupt apicobasal polarity, and you also disrupt normal cell shape because you're no longer correctly regulating the actin cytoskeleton. But the inverse is also true. So if you lose normal apicobasal polarity, so if you have mutation in a polarity protein, for example, that will disrupt apicobasal cell polarity, but it will also disrupt cell-cell adhesion. It will also disrupt normal cell shape. So you've got these three processes, and they're all sort of regulating one another. So you've got polarity, you've got cell-cell adhesion, you've got the actin cytoskeleton, and they're important to maintain normal cell shape, adhesion, and polarity, and to maintain tissue integrity. But if you disrupt one of these processes, then you disrupt all three. Now, just finish this part of the talk by mentioning that this type of invasion, EMT, is not the only type of invasion. It's not the only type of invasion or migration. There are other ways that cells can invade and migrate away from the primary tumor. So we've got the mesenchymal type of invasion and migration that I've spoken about earlier, where a cell takes on front-rear polarity, where it secretes these matrix metalloproteases. But there's also another type called amoeboid migration, Here, you don't really take, the cell doesn't have any polarity. It doesn't have front, rear, or apicobasal polarity. And it doesn't secrete matrix metalloproteases, so it doesn't break down those protein fibrils in the ECM. Instead, it uses its actomyosin cytoskeleton to actually squeeze through pores between these protein fibrils. And actually, amoeboid type of migration is the fastest migration that a cancer cell can use. And the third type is collective cell migration. This is where a whole clump of cells can invade. It can break off a tumor and invade together and migrate away together. Now, importantly, all of these different types of invasion and migration have been seen and observed in different types of human cancer. And also importantly, cells can actually switch the type of migration that they use as they migrate away from the primary tumor. So you might have a primary tumor where a clump of cells break off and invade collectively, and then it might start migrating away, and then eventually a cell breaks off from that clump of cells and migrates away in an amoeboid fashion. And then that cell might find itself in a very, very tightly knitted part of the extracellular matrix, and it gets stuck. So it switches to a mesenchymal type of migration, and that's where it starts to secrete the matrix metalloproteases. It can break down the extracellular matrix. It can continue its journey away from that primary tumor. So we'll move on to metastasis. So invasion is the first step of what's known as the metastatic cascade. So the metastatic cascade is a whole series of events that a migrating cancer cell has to undertake successfully in order to form a secondary tumor at a secondary site. Now, it's important to stress how metastasis and the metastatic cascade is a very inefficient process. The vast majority of migrating cancer cells will die at some point along this journey, some point along this cascade. The vast majority of cancer cells will die. Unfortunately, even if it's a very, very small proportion of cancer cells that successfully make it, you only need a very small proportion in order to form multiple secondary tumors at multiple secondary sites. So we spoke about the first step, and that is invasion. So we've spoken about how a cell loses polarity, breaks its junction, loses normal cell shape, can secrete these matrix metalloproteases, can migrate away, and it can migrate away using different types of migration, including collective, mesenchymal, or amoeboid. The next step in the metastatic cascade is intravasation. This is where the cell encounters the vasculature and it can intravasate into the vasculature. Once it gets into the vasculature, then it finds itself in the circulatory system, and it can be transported rapidly around the body. And they can transport the migrating cancer cell to distant sites in the body very rapidly. However, the vasculature is a very hostile environment, and the migrating cancer cell has to find a way of surviving within that hostile environment. It then has to find a way to extravasate and exit the vasculature at a certain point. Once it exits, it extravasates, it will find itself into a completely new place, most likely very distant from the primary tumor, and it has to survive in this new environment. And what it usually does is it inserts itself into an epithelium at that secondary site, and it forms what's known as a micrometastasis. And once it forms this micrometastasis, it's just one step away from completing the metastatic cascade, and that final step is known as colonization. This is where a micrometastasis is able to divide and survive and thrive and form a macroscopic tumor at that secondary site. Now that's sort of the metastatic cascade in a nutshell. Now I'm going to go into it in a bit more detail. So we've spoken a little bit about intravasation previously when we were talking about angiogenesis. So you've got angiogenesis bringing the vasculature into close proximity. The fact that they're very leaky means that it's very easy for these migrating cancer cells to intravasate into the vessel. However, once they find themselves in the circulatory system, as I said, this is a very, very hostile environment for these migrating cancer cells. And it's a hostile environment for a couple of reasons. The first is that you've got very, very high hydrodynamic shear forces in the circulatory system. If you think about cells that live in the circulatory system, they are very, very highly specialized to survive within the circulatory system, whereas a migrating cancer cell will not be specialized to survive in that circulatory system, so it can very, very easily be damaged and killed simply through the high hydrodynamic shear forces. Secondly, while it's in the circulatory system, it's sort of a sitting duck. It can very, very easily be attacked by cells of the immune system, such as natural killer cells, lymphocytes, and macrophage. So cells have to, these migrating cancer cells have to find a way of surviving in this very hostile environment, and they do find a way, and they do this by forming what are known as microemboli. What they do is they sort of absorb onto their surface platelets and red blood cells to form sort of like a shield that protects them both from the high hydrodynamic shear forces, but also from attack by the immune system. And you can see the importance of this protective shield in this experiment here. So here the scientists, what they did was they took a mouse and they took melanoma cells and injected those melanoma cells into the tail vein of these mice. And what they were looking for was, were these melanoma cells able to travel to the lung and form metastases in the lung? And they did this on two different types of mice. One mouse was a wild type, and the other had no platelets. It had a mutation which meant that no platelets were formed. And what they found was when they injected the melanoma cells into the wild type mice, these metastases formed in the lung very readily, many metastases formed. However, when they injected them into the mice that had no platelets, no metastases formed. And the reason why no metastases formed was simply because they didn't reach the lung. They died before they could reach the lung. And it shows the importance of those platelets in forming this protective shield around the migrating cancer cells, allowing them to survive within the circulatory system. In this case, the cells died within the circulatory system. Here is another experiment that shows pretty much the same thing, but here the mice don't have a mutation that means that there's no platelets. Instead, there's a mutation in an adhesion molecule called p-selectin. So p-selectin is how the platelet attaches to the cancer cell. Now, here you can see in a wild-type mouse, you can see the cancer cells in green, and they're surrounded by blue, and blue represents the platelets. In this scenario, they're mutant in p-selectin, and here you can see that they're no longer surrounded by blue. Instead, they're surrounded by red, and the red are the immune cells. So far, we've traveled quite a way through the metastatic cascade. We've had a tumor, it's progressed to become invasive, a cell is broken away from that primary tumor, migrated away, it's intravasated into the vasculature, it has found a way of surviving within the circulatory system. And the next step is for these migrating cancer cells to extravasate from the circulatory system into a new location in the body. Now, this extravasation can occur in a number of ways. So there's a brute force way in which a migrating cancer cell can extravasate, and there's also a more elegant way that involves a number of different adhesion molecules. Now, the brute force way is pretty simple. So you've got these migrating cancer cells, they're in the circulatory system, they're being pumped around the body, and then eventually a migrating cancer cell might get stuck in a blood capillary. So it gets stuck in the blood capillary, and then it starts to proliferate within that blood capillary, and then it proliferates to such an extent that the capillary ruptures, and that releases the cancer cells out of the circulatory system and into a new tissue in the body. A very simple way of extravasating. Now, the more elegant way involves a number of adhesion molecules, and in this way, the migrating cancer cell is actually mimicking how white blood cells extravasate. So white blood cells are very nomadic cells. They're constantly being pumped around the body, waiting for some sort of a signal, waiting for a signal that there's a location in the body where there's a location of inflammation or infection. And when they get that signal, the white blood cell will extravasate in the vicinity of that location of inflammation or infection. Now, white blood cells do this using a number of adhesion molecules, and it's sort of like a two-stage process. You get a weak adhesion, which is selectin dependent, and you get strong adhesion, which is integrin dependent, and that leads to the extravasation of the white blood cell. So this extravasation of white blood cells relies on selectins, which bind to specific oligosaccharides, which are present on the white blood cell, integrins, which bind to specific immunoglobulin superfamily members as well. So what happens if you have a site of inflammation? You get pro-inflammatory cytokine release. Those will then bind to cytokine receptors, which are present on a number of different cell types. And that will lead to those cells expressing and presenting on their surface the selectins, integrins, and immunoglobulin superfamily members. So when we think about white blood cells, so you've got a white blood cell, they're constantly being pumped around the body, waiting for some sort of a signal. There's a site of inflammation or infection. You get these cytokines binding to the cytokine receptors on the blood vessel wall. That will lead to the endothelial cells that line those vessel walls to express on their cell surface the selectins. So the next white blood cell that comes along will then bind to those selectins, and so bind to the side of the vessel wall. Now this will lead to a sort of like a rolling effect on the side of the vessel wall, because what you have is the selectin adhesion is a very weak type of adhesion, and that combined with the high rate of blood flow will lead to a rolling of the white blood cell along the vessel wall, and that will trigger the strong adhesion step. So it's important to say that these selectins, I spoke about selectins previously when I was talking about platelets and how they bound to the migrating cancer cell. But endothelial cells also express P-selectin and E-selectin. But importantly, they only express P-selectin and E-selectin when they become activated by these cytokines. So normally they don't express them on their cell surface. They only express them when they have a trigger, when they've been told that there is a site of inflammation or infection in the vicinity. So as I said, these white blood cells, they will bind to the vessel wall when there's a site of inflammation or infection, because the selectins are presented on the surface of these endothelial cells. The high rate of blood flow will lead to the rolling of the white blood cells, and that will trigger the integrin-dependent step and the extravasation of the white blood cell. So what happens is the rolling will trigger the white blood cell to express an integrin. So for example, LFA1, that will bind to an immunoglobulin superfamily molecule called ICAM1, and that strong adhesion will lead to the extravasation of the white blood cell. And you can see how the white blood cell will extravasate specifically in the vicinity of the site of inflammation or infection. Now, the integrin family of adhesion molecules and the immunoglobulin superfamily of adhesion molecules are massive families, but we're only really interested in a couple of them. We're only interested in VLA4 and LFA1 because they bind to VCAM1 and ICAM1, and VCAM1 and ICAM1 are expressed on activated endothelial cells. So just like with the selectins, VCAM1 and ICAM1 aren't usually expressed and presented on the surface of the endothelial cells. It's only when they get a signal that there's a site of inflammation or infection that both the selectins and also VCAM1 and ICAM1 are presented, facilitating both the weak and the strong adhesion that allows these white blood cells to extravasate in the vicinity of the site of inflammation and infection. So how do cancer cells mimic this? Well, they simply present on their cell surface the specific oligosaccharides, the integrins that facilitate both the weak and the strong adhesion. So they basically mimic white blood cells. And this is the reason why if a person has circulating cancer cells in their circulatory system and they also have a site of inflammation or infection, metastases will form preferentially in those sites of inflammation or infection, because all the signaling is there. The signaling that promotes white blood cell extravasation will also promote the extravasation of the cancer cell in the vicinity of that inflammation or infection. So, cancer cells can also, they don't have to rely on pre-existing sites of inflammation or infection to extravasate, because they can be sources of cytokines themselves. They can secrete cytokines, which will bind to the cytokine receptors on the endothelial wall, and that will lead those endothelial cells to present on their cell surface both the selectins and the immunoglobulin superfamily members that will facilitate both the weak and the strong adhesion. And that means that cancer cells can extravasate anywhere. They don't have to rely on pre-existing sites of inflammation or infection. So cancer cells can extravasate in a variety of ways, including the brute force way, including using the signaling from pre-existing sites of inflammation or infection, or they can be sources of cytokines themselves and extravasate anywhere in the body. So once they have extravasated, they will find themselves in a completely new location in the body, most likely very far away from the primary tumor. And their response to finding themselves in this completely new environment is to insert themselves into an epithelium almost immediately. Now, this happens through what's known as mesenchymal to epithelial transition. It's the opposite of EMT. So here the cells lose mesenchymal characteristics and instead take on epithelial characteristics. And really what they're doing is because most cancers are epithelial in origin, what they're really doing here is they're just reverting back to taking on the epithelial characteristics that they had in the first place. Now, it's thought that the reason why that this almost immediate insertion into an epithelium at the secondary site happens is because of what I'm about to tell you now. So what I'd like to do now is go back to the primary tumor and have a think about what happened at the primary tumor site. So at the primary tumor site, that primary tumor has spent decades acquiring mutations to allow the tumor to progress toward malignancy. Spent decades acquiring mutations in oncogenes, tumor suppressor genes, deregulating pathways, etc. But not only has it spent decades mutating its own genome, it's also spent decades remodeling the local microenvironment. It's attracted different cell types, it's induced those different cell types to secrete factors that promote tumor proliferation, tumor survival, tumor progression, and invasion. So when a tumor progresses to become invasive, what that tumor cell has when it invades is it has both intrinsic factors, mutations within its genome that promote tumor invasion, but it also has extrinsic factors that promote invasion, this very highly permissive microenvironment at the primary tumor site. Now, let's fast forward now to this tumor cell that has just extravasated at a distant site in the body. It's extravasated at a completely new location, and this new location has not been remodeled by a tumor. This new location is not highly permissive to tumor survival and proliferation and invasion. This new location is a very hostile location. It's full of anti-growth factors, it's full of death factors. And the tumor cell still has these intrinsic mutations that promote invasion, but it's lost these extrinsic factors that promote invasion. And for that reason, as soon as it extravasates and as soon as it finds itself in this new hostile environment, it will insert itself into a tissue as soon as possible, because otherwise it will simply die. So that brings me to the final step of the metastatic cascade, which is colonization. So metastasis has two phases. The first phase is the physical dissemination of cancer cells from the primary tumor site to another site. And that's basically what I've spoken about so far. But the second step is by far the most complex step, both in terms of the cancer cell successfully colonizing and also in terms of of scientists successfully understanding how colonization works. Because the second step is how that cancer cell adapts to this new environment and is able to successfully survive within this new environment. Most cancer cells can successfully disseminate but fall on this final hurdle. They will die in this final step because they are unable to adapt to this new location. So when a micrometastasis forms, two things can happen. Either the micrometastasis will form and then the cell will die because it cannot successfully adapt to this new location, or the micrometastasis will remain dormant. And it can remain dormant for an extended period of time. It can remain dormant for decades. It can remain dormant for a lifetime. But in many cases, the dormant micrometastases will suddenly flare up, just suddenly. After decades of not doing anything, they will suddenly flare up. And they're thought to be able to flare up after a long period of dormancy because they will have solved the problem of being able to survive within this new environment. So it's likely that when a micrometastasis forms, it will be poorly adapted to this new location. So it will have to adapt. It will have to adapt both in its intrinsic capabilities, so it will have to acquire new mutations in order to colonize, but it also have to adapt the local microenvironment. So if you remember, the primary tumor spent decades remodeling that local microenvironment to make it permissive to tumor growth and survival. Well, at the secondary site, that cancer cell that has formed that micrometastasis will have to do that all over again. It will have to attract different cell types, induce those different cell types to secrete factors that promote tumor survival and growth. And so the cancer cells have to do both of these things. They have to acquire additional mutations and also remodel the local microenvironment to successfully colonize. So these migrating cancer cells, as I said, they arrive at a completely naive, fully normal environment. And so the cancer cells will have to induce new cell types to attract these new cell types and induce them to secrete factors that promote tumor survival and growth. And this is why colonization is so difficult to understand, because these interactions involve multiple different cell types and they're constantly changing. It's very dynamic. As the cancer cell acquires new mutations, the signals coming from the tumor will change, but also as different cells are attracted, different stromal cells are attracted to the tumor, as the extracellular matrix is being remodeled, then the signal coming from the stroma will also change. So not only is it complex because it involves many different cell types, it's also complex because this sort of talking between these cell types is constantly changing. As the tumor develops, as different cell types are attracted to the tumor, that the conversation going on between these different cell types changes over time. So the tumor arrives at this very hostile environment, forms this micrometastasis, and then it embarks upon this long process of remodeling the local microenvironment, but also acquiring new mutations. And that will allow it to colonize and form a macroscopic tumor at the secondary site. But this isn't the end of the story, because once a secondary tumor forms, that secondary tumor itself can then progress to become invasive itself, and cells can break off that secondary tumor and migrate away from that secondary tumor and can form what's known as tertiary tumors in completely new locations in the body. Or they can actually undergo what's known as reverse migration, which is where the migrating cancer cell will break off the secondary tumor and migrate back to the primary tumor. And this reverse migration is actually very attractive to a migrating cancer cell, because that cancer cell does not have to undergo the entire metastatic cascade. Because if a cell breaks off the secondary tumor and then goes off to form a tertiary tumor, it will have to do the entire metastatic cascade. You have to travel to a distant site, have to form a micrometastasis. It will then have this long period of dormancy where it remodels the local microenvironment, etc. But if it undergoes reverse migration, it doesn't have to do that. It just has to travel back to the primary tumor, and it doesn't have to undergo this period of dormancy. It doesn't have to remodel the local microenvironment, because at the primary site, the tumor site is already very permissive. The local microenvironment is already very permissive. And so it doesn't have to undergo the entire metastatic cascade. But this reverse migration can dramatically change the behavior of the primary tumor, because those reverse migrating cancer cells will bring with them new mutations, and that can mean that the primary tumor becomes even more aggressive than it was before. So with that, I will finish, and I will say thank you for listening. Hopefully you understood everything I said.
Excellent, thank you so much. Cool, I will just stop the recording so people can then ask questions. I hope that stopped it. Stop recording. Stop.